Good morning, everyone. So here we'll be discussing about some developments in the science and technology part from exam point of view. So recently, some developments have made. Number one is humanity's last exam. It's a new frontier in AI benchmarking. So, it is proposed to test artificial intelligence systems at their highest potential by creating an exam that pushes the boundaries of their cognitive abilities and decision making. And the idea behind this exam is to benchmark AI against tasks that demand not just technical precision, but deep understanding, ethical reasoning, and creativity skills traditionally associated with human intelligence at its peak. So here actually the it is given the exact definition that it is a multimodal benchmark design to push the boundaries of AI evaluation, and it represents a diverse and exhaustive collection of 3000 challenging questions across over 100 subjects, encompassing fields such as mathematics, humanities, and the natural science. So, unlike traditional benchmarks that primarily feature text based questions, the dataset incorporates diagrams, images, and other formats to assess an AI's ability to process and understand multimodal information. And the creation of humanity's last exam was a global collaborative effort. Nearly 1,000 subject experts, including professors, researchers, and graduate degree holders from over 500 institutions across 50 countries, contributed questions. So, this diverse input ensure the benchmark reflects a wide range of academic disciplines and perspective. So, from examination point of view, questions may be arise from the ethical implications, awareness of technological advancements, then the society and policy making, and ability and their link to their contemporary government and all. So, I have given one question that is, with reference to the concept of humanity's last exam, consider the following statements. Number one is, it is a global AI benchmark designed to test ethical reasoning and adaptability. Number two, it focuses exclusively on programming and computational skills of AI. So, which of the statements given above is or are correct? So, the correct answer is A, that is one only, that it is a global AI benchmark designed to test ethical reasoning and adaptability. After that, number two, antibiotic resistance breakthrough, so targeting plasmids for new treatments. And international collaboration has achieved an important breakthrough in understanding the genetic mechanism that allow bacteria to build resistance to drugs. And bacteria have multiple defense mechanisms they can use to build resistance to antibiotics, one of the major problems facing public health globally. And one of these mechanisms involves plasmids, so which is small DNA molecules in bacterial cells and which have their own independent genome and carry antibiotic resistance. And if it can work out like the roles of plasmids play inside the bacteria, then they can use the information to develop a new generation of therapeutics that can target drug resistance infections. So, research have been made using a model plasmid called RK2 that is used globally to study clinically relevant plasmids that transmit antimicrobial resistance. And the initial focus was a molecule called COR-B, which is essential for plasmids to survive within their bacterial host. So, this DNA binding protein was previously known to have played a role in controlling gene expression, but how this happened was unclear. So now this approach could complement existing antibiotics, restoring their efficacy against resistant infections, and it may also reduce the selective pressure that drives resistance evolution. And coupled with better antibiotic plasmid targeting strategies, offer a promising path forward in the global fight against antimicrobial resistance. So, talking about genome editing, we must know about CRISPR case technology. So, it stands for Clustered Regulatory Interface Short Palindromic Repeats, and it is a natural defense system found in bacteria where it protects against invading viruses by targeting and cutting their DNA. So, what are case proteins actually? So, these are CRISPR associated proteins, for example, case 9, that act as a molecular scissors cutting DNA at specific locations. So, in simple terms, we can say that CRISPR case is a precise and programmable DNA editing tool that mimics this natural bacterial system for use in genetic engineering. So now let's see how it actually works. So scientists design a short RNA sequence complementary to the target DNA sequence, and this is called guide RNA, that is gRNA, and it guides the case protein to the exact location in the genome. And then see case protein actually, for example case 9. So the case protein binds to the gRNA and cuts the DNA at the targeted site. And then DNA repair. So after the cut, the cell repairs the DNA in one of two ways. So number one is non-homologous end joining. So it repairs the cut with errors leading to gene disruption. And two is homology directed repair. So it uses a template to insert or replace specific DNA sequences. And now let's practice some questions from exam point of view. So number one is, which of the following is associated with the spread of antibiotic resistance in bacteria? So the correct answer is B, that is plasmid. Then number two is, the recent scientific breakthrough in targeting plasmid is related to. So the correct answer is B, that is antibiotic resistance. After that, question number three, CRISPR case technology is primarily used for. So the correct answer is D, that is all of the above. So DNA sequencing, editing genetic material, and whether it's plasmid elimination. That's all. Thank you.